the discovery of DNA. It can easily be said that DNA is the most essential molecule of life. Like a computer's hard disk, DNA holds encrypted all the information that a living creature needs to exist. In fact, there are millions of tiny hard disks scattered in a human's body, one in every cell. In analogy, although all humans carry the same folders and even the same files, the content of these files, namely the information that is written in them, is different in each and every one of us, making us individuals. The language that is used contains only 4 letters, which are surprisingly enough for the description of the most complicated mechanism on Earth, the human being. Hidden as it is inside the nucleus of a cell, surrounded by 2 membranes, the nuclear and the cytoplasmic, DNA was the last of the cell's building blocks to be discovered. While proteins were known since the 18th century, it was not until 1869 that a 25-year-old Swiss doctor discovered a new substance with unusual properties that today is known as DNA. This was Friedrich Miescher. After graduating from the Basel University's medical school, Miescher moved to Tübingen to be trained as a scientist. The field that he was most interested in was physiological chemistry, what could today be called biochemistry of life. In a laboratory that was placed at the former kitchen of the Tübingen castle, Miescher was studying the chemical components of animal cells. As a model system, he used leukocytes, namely white blood cells, because they were easy to find from the local surgical clinic. Surgical bandages contained pus, which in turn contains large amounts of white blood cells. As he was studying proteins and lipids he isolated from these cells, he mentioned a strange substance that was quite different from the cell components known until then. It could be precipitated by acids and redissolved by adding alkaline solutions. Based on his observations, Miescher realized that this novel substance came from the nucleus and named it nuclein. In the years to come, Miescher managed to improve the isolation protocol and to obtain the first comparatively clean preparation of DNA. He also uncovered the elementary composition of nuclein. He found carbon, hydrogen, oxygen, nitrogen, and a large proportion of phosphorus. Therefore, he suggested at first that nuclein might serve as a storage of phosphorus. Miescher's supervisor was skeptical of his data and therefore repeated Miescher's experiments. Only after he managed to replicate Miescher's results on nuclein did he allow the publication of his manuscript. 2 years after the discovery of nuclein, in 1871, Miescher's work was finally published under the title Über die chemische Zusammensetzung der Eiterzellen, which means on the chemical composition of pus cells. Miescher was confident about the importance of his discovery and therefore he continued working on nuclein, but changed the source. He used salmon sperm to isolate a much larger quantity of nuclein and of better purity. Working with salmon sperm DNA, Miescher noticed that it should be a multibasic acid and that it must have a high molecular weight. Miescher also came very close to uncovering the role of nuclein, as he suspected an involvement in fertilization. Miescher died at the age of 51 due to tuberculosis without receiving the appreciation he deserved for his tremendous discovery and without fully realizing it himself. The term nucleic acid, initially Nukleinsäure, was attributed to nuclein by Richard Altman in 1889. In 1938, Levin uses the term desoxyribonucleic acid for the first time, while the abbreviation DNA was first used in 1944. This is actually the beginning of molecular biology.